Let's jump right in. In this explainer, we're diving into how the Linux command line acts as the absolute backbone of modern computational biology. Because let's be real, if you're handling biological sequence data today, the terminal is where the actual magic happens. Look, we've all been there, right? You try to open a massive next-generation sequencing file and boom, your graphical interface completely freezes. It's incredibly frustrating. The terminal is basically your magic bullet here. It completely strips away all that visual bloat, meaning you can effortlessly automate and process thousands of files without breaking a sweat. The biggest takeaway to keep in mind is that almost all the heavy-hitting genomic tools, whether you're talking GATTech or SAM tools, are built specifically for the Linux environment. It gives you the portability, the strict reproducibility, and just the pure, unadulterated performance that modern science actually demands.
So, stepping into the terminal, it means taking total control of your environment. You start by typing pwd, print working directory. Think of it as your compass. It anchors you, telling you exactly where you're sitting in the file system before you make your next move.
From there, LSLA is essentially your X-ray vision. Slap on those -LA flags, and you instantly get this deeply detailed list of your directory. We're talking hidden configurations, absolutely critical file permissions. Suddenly, your environment has literally zero mysteries. Okay, so you've got your bearings. Now CD, or change directory, becomes your personal teleporter. It lets you seamlessly jump from your home folder straight into the deepest, most complex project structures in a split second. And to tame the sheer chaos of incoming genomic datasets, mkdir is your go-to organizational tool. It instantly spawns new directories, keeping all those sequences clean, tightly structured, and perfectly primed for analysis.
What's really wild is how these core file manipulation commands let you sift through vast amounts of data without ever double-clicking a single file. You're copying with CP, moving stuff with MV, or peeking into massive DNA sequences using head or tail. A quick word of warning now, definitely tread lightly with RMRF. It will forcefully wipe directories out of existence without any safety net. Now, to really grasp the true power of all this, we have to talk about the Unix philosophy. The whole idea is beautifully simple. Make each program do exactly one thing, but do it incredibly well. And, crucially, treat all data, even our complex biological DNA sequences, as simple text streams. This means we can link tiny, super efficient tools together to pull off some incredibly complex operations. And this philosophy, it gives birth to the pipe. By just using a vertical bar, we can chain these isolated tools into a massive, powerful assembly line. Picture this. We stream a raw FASTQ file, filter for sequence headers using grep, and send that directly into WC-L to instantly count the total reads. That is 3 simple commands delivering one massive biological insight. Once you have those basics locked down, we can unlock advanced stream manipulation. Tools like awk and sed are basically surgical instruments. You can use awk to isolate specific single nucleotide polymorphisms in variant call format files, or use sort and unique to organize genomic coordinates and count duplicate sequences entirely on the fly. It is just so fast. Take a specific use of sed replacing headers, for example. Sure, at first glance, the syntax just looks like a random string of characters, right? But a single line of code can instantly transform a bulky FASTQ file into a streamlined FASTA file just by manipulating text patterns. No heavy software required here at all. It is just pure, elegant text surgery. So, zooming out, these foundational command line skills become your essential launchpad for enterprise-level research. Whether you're managing software packages with Bioconda to guarantee lab-to-lab reproducibility, or orchestrating automated, massive workflows on high-performance computing clusters using Nextflow or SnakeMake, Linux is the engine making it all possible. Sure, graphical interfaces might feel a little safer at first, but let's be honest, they're a huge bottleneck dear research. The terminal isn't just another tool, it is the native language of modern biology. So, the next time a massive sequencing file lands on your desk, are you going to risk freezing your screen again, or will you fire up the terminal and truly take control of your data?